church, and I dressed up for presentations.
And I'm Brandon Coyne, and you call me B-Swizz.
All right, 600,000 is the number of people who die a year of malaria. So we're talking about a technology that could potentially solve that problem.
And the way to solve the problem is by using gene drives. And gene drives are a way to change the gene pool of an entire species. And by doing that, we can engineer mosquitoes to not spread malaria. CRISPR is a gene drive, and it is part of the bacterial immune system. And how CRISPR works is that a virus must infect a bacteria, and then the virus inserts its DNA in the bacteria, and then the bacteria takes the DNA and uses it for defense. And then CRISPR comes in and it locates the virus's DNA, and it finds it by locating sequences, and then it cuts it out.
So gene drives aren't a new idea. They're something that the idea came like in the 1970s, I think, but they didn't have CRISPR back then. And so now that they have CRISPR, which, like Brandon said, can locate a specific gene and then cut at that spot in the genome, they could apply the idea of gene drives. So this kind of shows how. So they put CRISPR in with the specific gene that they want to change. And so, I mean, you get a gene from your dad, you get a gene from your mom. And so the idea of a gene drive is you want not 50% of the population to inherit the gene, but 100% to inherit the gene. And so if we look at the blue mosquitoes, we're saying the mom, let's say, is the blue mosquito, and that's the one that wouldn't pass on malaria. And usually it would be 50-50. But if we have CRISPR in the gene, then it'll go in and cut the dad's DNA where the malaria gene isn't. And then to repair itself, the DNA is going to look at the homolog, which is going to be the mom's DNA, and they're going to copy that. So then both pairs are going to have the the gene for malaria resistance. Oh, that's pretty much the same thing, so. Okay.
Some gene drive limitations. It cannot be used in crops because we engineer the seeds ourselves. It cannot be used in humans and animals because the reproduction takes way too long. And in cattle, it cannot be used because they're tightly breeding systems, and there will not be very much gene mixing.
So the other thing on the slide is bacteria, and so it has to happen through sexual reproduction. So bacteria doesn't fit that. This is where it would be applicable and helpful. In invasive species, the idea is that the gene that they transfer through the whole species would be one like seed, like make all the eggs infertile or something. Like that's a lionfish and it's not native in Florida. And so if they could go through and make every single fish female, then obviously the population isn't gonna last. And then the mosquitoes. So if they could, there are like plenty of diseases that mosquitoes pass on that kill people every year. And mosquitoes reproduce so fast that it could definitely be done. They think in a year, all mosquitoes could be malaria resistant. Oh, and then all the bugs that develop pesticide resistance, and so we have to keep getting like more and more chemicals and stuff like that, could engineer the bugs to not be pesticide resistant. Or engineer, like the bees, they could change it so that the bees are all pesticide resistant. Okay. This is a quote from someone who worked in a lot of the studies done on this. because they're really afraid that if this technology gets out, it's not being applied yet because if, like with the lionfish, if they make every fish female, but that somehow spreads to a native population, I mean, because all water is connected, then that's going to wipe out the native population and the invasive population. Also, in species where you have a non-native and a native species that are closely related, then if it gets into the native species, then it's going to wipe that out too. And so there's a lot of dangerous parts to this technology.